The first stage of tooth development is the initiation stage. In the 6th week of development, the embryo forms an indentation called the stomaedeum. This section is also called the primitive oral cavity and is composed of epithelial cells. Around week 7, the layer of epithelial cells within the stomaedeum separate into the dental lamina, which forms the primary or baby teeth, and the vestibular lamina, which become the permanent teeth. The focus of our project was on the primary tooth development. Within the dental lamina, there are epithelial and mesenchymal cells. The top layer of epithelial cells will send signals BMP and FGF to the mesenchymal cells. BMP and FGF induce transcription factors within the mesenchymal cells. These include MSX1, MSX2, DLX1, DLX2, and PAX9. Reciprocal induction occurs and the mesenchymal cells send signals to the epithelial cells. These signaling molecules are BMP4, activin, and FGF. Stage 2 is called the bud stage. Wnt and TNF promote activation of ectodisplacin receptors, which regulate dental placoid development. The placoid releases BMP, FGF, Wnt, and SHH to induce proliferation of the epithelium, also known as budding, while the mesenchymal cells condense. FGF and RUNX2 are transcription factors that signal the transition from the bud stage to the cap stage. Stage 3 is known as the cap stage. In this stage, the mesenchyme releases BMP to form the enamel knot. This signal also causes transcription factor P21 to activate, along with the Edgar receptor. The enamel knot signals both the epithelial and mesenchymal cells with FGF, WNT, BMP, and sonic hedgehog signals. Cervical loops form within the epithelium in response to sonic hedgehog signaling from the enamel knot.
Bell stage is initiated by the formation of the secondary enamel knot. This stage is known as Bell stage because this is when the cervical loops extends outward and the enamel knot pushes up to form a bell shape. The primary enamel knot signals for the secondary enamel knot to form along with the signaling factors from the mesenchymal cells. Signaling factors of the mesenchymal cells include Wnt, BMP, and FGF. Here, the enamel knot induces signals such as SHH, BMP, FGF, and Wnt to regulate the patterning of the tooth crown. Then the epithelial cells starts to form the shape of the crown. The signaling factors of the enamel knot regulates the enamel epithelium to pinch off from the dental lamina surface. The enamel epithelium then differentiates more and is now ready for crown development. Apposition stage is the crown development stage. This is the enamel epithelium after it pinches off from the dental lamina. The blue lines represent the enamel epithelium. The outer enamel epithelium, or OEE, is on the outside of the inner enamel epithelium, or IEE. The IEE is on the inside surface. The IEE is lined with stratum intermedium cells. These cells are high with alkaline phosphatase activity and help with enamel formation. Between the outer enamel epithelium and the stratum intermedium are the stellate reticulum cells. These are star-shaped cells. These cells are lost after enamel form. In the next picture, we'll look up closer at the inner enamel epithelium, or the region shown in the box in this picture. apposition is initiated by the signaling of the enamel knot. The enamel knot induces BMP, FGF, SHH, and Wnt to signal odontoblast cells to induce the inner enamel epithelium to elongate into tall columnar cells of ameloblasts. Odontoblasts are cells that will form dentin, and ameloblasts are cells that will later form enamel. Here, ameloblast induce odontoblast to form dentin. Signaling factors such as TGF-beta, NFIC, Wnt, and FGF induce odontoblast to form dentin. As dentin is produced, which is shown in red, it induces ameloblasts to form enamel. Enamel is shown in the yellow region in this picture. This is an example of reciprocal induction. This picture shows the full development of a tooth crown at 3 months of age for an individual. This picture is zoomed into the end portion of the crown. An important component of root development is Hertwig's epithelial root sheath, or HERS. HERS is formed from the outer enamel epithelium and inner enamel epithelium located at the neck of the crown and grows in apical direction. HERS outlines the future root and is responsible for size, shape, length, and number of roots. Here, HERS secretes laminin 5 to induce the dental papillae to form odontoblasts. Odontoblast is induced by TGF-beta, NFIC, Wnt, and FGF to form dentin. Here, dentin is shown in red and odontoblast cells are to the right in brown. As dentin forms, HERS disintegrates into cells called rests of Malassez to allow for dental follicles to come in and interact with dentin. HERS secretes SHH, patched, and Gli1 to induce dental follicles. The interaction of dental follicle and dentin induce dental follicles to form cementoblasts. Cementoblasts are then induced by TGF-beta, NFIC, Wnt, and FGF to form cementum. This cycle of cementum formation continues and grows downward by having HERS induce odontoblasts to deposit dentin. HERS then breaks off to allow dental follicle to penetrate and deposit cementum on root surface. Then the cycle starts again, each time HERS dissociates further. As HERS disintegrates, dental follicles form collagen fibers. These fibers are the periodontal ligament, which attaches to the root surface and grow into the periodontal space. As the root develops and elongates, the crown differentiates upwards toward the epithelium. The tooth erupts at around 6 to 10 months of age. The root continues to form and completes at 2 years. Tooth development is critical. Some abnormal growth occurs at HERS signaling. If HERS is interrupted too early, then the dental pulp and periodontal ligament will connect without dentin. If HERS fails to be interrupted at the correct stage, then there's no formation of cementum, and the exposed cervical region of tooth will cause sensitivity. TGF-beta BMP signaling is an important signaling pathway for tooth development. TGF-beta ligand binds onto the type 2 and 1 receptors. This then undergoes the phosphorylation of rSMADs. RSMADS is a mediator. It then goes on and binds onto SMAD4. This complex then translocates into the nucleus, where it regulates the expression of target genes. TGF-beta BMP signaling is needed for cementum formation, odontoblast differentiation, periodontal ligament cell proliferation, and is also expressed in HERS.